So now we're at the moment we have waited for, at least I've waited for. We've built up our understanding or reviewed our understanding of inheritance. We built up our understanding of selection. We talked about the adaptive landscape. Now, and we've always been in shallow time. Now we're ready to go in deep time with the elements in our course. And what is going to be the unifying concept that enables us to pull everything together? That's a question. What is it, Gary? What is our unifying? Yeah, you weren't ready for me to call on you. So what is our unifying concept? I once went to Davis and I said, what is the most important unifying concept in evolutionary biology? And there was this very much like this response. Very much this response. Now, if Sewell Wright were in the room, or Russ Landy were in the room, they would stick up their hand and I would call on them. What would they say? I would hope they would say, the adaptive landscape, Steve. That's the unifying concept, and that's why our lecture is on the evolution on the adaptive landscape. Okay, so here's our thesis. Number one, always three, right? Three simple things. Models for adaptive radiation can be constructed with quantitative genetic parameters. Yes. The use of quantitative genetic parameters allows us to cross-check with the empirical literature on inheritance, selection, and population size. Now, I'm making this a point because later in the course we will often use what I call generic models of adaptive radiation that have no connection to quantitative genetics. And so there's no way to cross-check parameters the way we're going to do a little bit today. Okay, and then here's a conclusion we'll reach. Stabilizing selection and peak movement appear to be necessary, but may not be sufficient to account for evolution in deep time. So that's a big conclusion, but that's what we're going to work for today. And this lecture is even worse than some of the others because it's packed with stuff at the end, so I'm going to have to skip along lightly so we can spend a little more time being on the same page for this last point. Okay, here's what we're going to do. We're going to start with evolution without selection. That is to say, we're going to start with genetic drift. We're going to then talk about univariate evolution on a landscape with a single stationary adaptive peak. And then we're going to go to bivariate two-trait evolution on a two-dimensional adaptive landscape, but with a single peak. And then we're going to allow the peak to move, and we're going to talk about why we're going to have to make the peak move. And then we're going to actually look at some data, and we're going to say what happens when we try to explain actual data with these various models of adaptive radiation that we've introduced. So that's where we're going. Okay, so let's start with a simple case. Let's start with a case of I'm out of. Oh, I'm dying. Where's Joe? I want to blast him away. Really shutting that door. He doesn't know we're waiting for him. So what about that pointer of yours, Joe? Can I borrow that? Under that black thing? I don't know. That? Oh, that's a horrible thing. So it's too confusing. Ah, jeez. What? Where's your pointer? Can I borrow that? What? It's too complicated for me. Oh, maybe it's not too complicated. You like green better than red? Yeah, this is. I promise. I swear to the class that I will return your pointer. Okay, here we go. Okay. Evolution without genetic drift. So here's an interesting fact. Let's suppose we had a normal distribution whose variance was G. And we took independent samples of size n sub e from that population. What would be the variance among the means of those samples? Huh?
Can you say that again?
Suppose we had a normal distribution with a variance of G, and we took repeated samples from that distribution of size n sub e. We calculate the means of those samples. What would be the variance among the means be? I think I said it enough. That's right. That's right. And the reason that's useful to us is it tells us that if we wanted to start with a single population and in the next generation draw a random sample of parents and of population of size n sub e and look at the variance among those replicates, that we have an expression from that just drawn from this elementary sampling law. And so when we look at the prediction, look at the variance among a set of replicates that are evolving by drift, at any generation in the future, t, the variance among the means is going to be g time, g divided by n multiplied by t. So the variance is going to expand in a very predictable way. And so here's the random walk, the genetic drift of a single population. But we have this lovely expression. Oh, and the means are going to be normally distributed too. A bonus with a variance given by that very simple expression. So that a set of 100 replicates through time is going to look like this. So now I have to pause for a minute. So in the computer exercise we're going to give today, I'm going to give you the R script for a figure like this, and I'm going to ask you to animate the figure, and I'm going to give you the script from another program. So you're going to have to meld the two sets of scripts so you produce an animation. And if you're really, if you've got enough time, you can put red balls on the ends of the diverging populations, as you'll see in the animation. So I have to do this quickly here. Like this. This isn't 100 population. Oh, jeez. I forget. Lord. Yes. God, leave me. Didn't I check the box back there? Oh, I turned it off. That's what happened. All right. It's painful to watch me up here, I know. It's amazing I can do anything, isn't it? Voila. Maybe 25 populations. Okay, so there's replicate 2. Your results may vary depending on the size of how many replicates. Oh, I guess it's just, well, anyway, this is, anyway. Okay, you get the point. All right. Okay, so a simple model for adaptive radiation by drift. Very small effective population size. We can, in the animation that I showed you, varies n sub e, and you can see with bigger n sub e, you're going to get less divergence as a function of time in a nice linear way. Okay. Now let's look at some models with selection. And in particular, let's look at a single adaptive peak. And there are results due to Landy in 1979 that give us this result. This is familiar to us, right? So this is our expression for, this is the multivariate Breeder's equation. Here we're substituting another meaning for beta. And what you can show is that the change in, when you're evolving by this equation on the adaptive landscape, the change in the log of population mean fitness is given by this expression, And that expression is always positive. So that's telling you that on a surface, the general tendency, on a surface with topography, the general direction of evolution is uphill on the adaptive landscape. Okay? And so not surprisingly, if we have an Ornstein-Uhlenbeck process, which we're going to describe, which is described by, for example, a single Gaussian peak, and we're looking at the evolution on that, and we're going to define what that process is. What happens each generation is that the change in mean, we could move z bar t over here, then you've got delta z bar. It's given by this term, which is, so there are two things going on. There's a struggle between two things. This term describes the tendency of the mean to evolve towards the adaptive peak, uphill on the surface. This term, this symbolizes a draw from a normal distribution with a mean of 0 and a variance of g over n sub e. That, we showed in the previous slide, is the contribution of drift to the mean of the population each generation. And it will, in general, be away from the adaptive peak in one direction or to the left or to the right. So what happens is that the population will eventually reach an equilibrium under drift and stabilizing selection, well, directional selection towards the peak and drift away from it, so that these two processes are equal. So in general, we expect there to be, among a set of replicate lines, there will be a variance among those lines at some point t, and that variance is given by this expression here. Now, and I'm just going to point to the algebra here and point out an interesting thing. If we look at this, and we're looking at it, right? See this negative sign? That tells us that this is a negative exponential expression, right? And t is ever increasing. So what happens to this part of the equation as time increases? It becomes smaller and smaller, doesn't it? And so this part of the expression in this kind of brackets disappear, and so we reach a stable, we're going to reach a stable variance among the lines given by this part of the expression here. So it's not going to be ever expand. So I forgot to tell you that the blue line was the 95% confidence limits for the normal distribution that expanding. That's not what's going to happen here. We're going to get a different expression, a different picture, and the picture is going to look like this. With an Ornstein-Uhlenbeck process, a model of selection of stabilizing selection towards a single peak, the variance rapidly reaches we rapidly reach an equilibrium variance under most parameter values. And so a single population is going to do this, and replicate populations are going to do that. There's going to be bounded evolution, essentially, inside, and I've shown that bound with the 95% confidence limits, but you could show it in other ways as well. I'm not going to show you animation 2. You can, that's so you can have some fun later. Right, Chris? All right. Let's look at another possibility. So this is also, let's look at just the approach to the equilibrium. So we're going to, this is a deterministic response to a single Gaussian adaptive peak in 2 dimensions. Oh, let's go, sorry, we're going to go to two dimensions. I just did that bonk. Bivariate evolution about a stationary peak. And we're going to look at evolution with constant G matrices towards that peak. And we're going to see the effect of the shape of the G matrix on the trajectory. That's what we're going to be looking at here. We're going to throw away the stochastic. We could have a drift term, and we're just going to throw that away so we can look at the effect of the G matrix on the approach to selection. Now, this is something we looked at earlier, just in single generation, but now we're going to put populations out in a circle around an adaptive peak, and this is, so here's in red, this is the adaptive landscape. There's no omega plus P 1, 2 term. It's equal to 0. And we're to watch populations evolve towards the peak. Now, if the G matrix here in green, I'm sorry about the color scheme changing, if the G matrix is green, the trajectories are directly towards the surface. In general, though, we can anticipate the approach to the optimum under deterministic Gaussian evolution is going to be affected not only by the shape of the G matrix, but by the shape of the adaptive landscape. And that's what we're going to look at in the next few slides. So for example, whoops, let me back up here. Oh, drat. I have to do this in animation, so I'm going to have to do it. Trying to avoid playing these today. But there's no escape. Okay, here's a case in which we have an ellipsoid adaptive landscape, but circular G matrices. Now, we can say with confidence that it's the ellipsoid shape of the surface that's causing these trajectories, right? Because we just looked at the case in which the adaptive landscape was circular and we had trajectories directly towards the peak, right? And why are the trajectories so I'm showing you arrowheads every 50 generations. Why are the trajectories so very rapid? See, the first interval between the first arrowhead, the population moved that entire distance. Why is that?
Selection can be the strongest the farther away you are from the adaptive landscape.
You got it, Dave. That's right. And I showed you that expression, right? The further away, the strength of beta is directly proportional to the distance from the peak. Okay, so that's one case. Look at this case. This is like the reciprocal. Now the adaptive landscape is circular, but the G matrix is cigar shaped. Beep, beep, beep. So what do you see in the initial portions of evolution? That's right, but what is that direction that the initial portions of all the trajectories are on? What do we call that along the long axis of the G matrix? The leading eigenvector, G max, right? That's what Whitlock was talking about. So you're seeing rapid evolution initially along G max, and then a period of slow evolution wherever you start in the direction of G min, which is the second eigenvector, the direction in which there's minimum genetic variance. And you can see that it's very, very slow primarily because of the compromised amount of genetic variation. Okay, this is the case we started with. And here's one in which we'll do both. You can capture this, I think, later. Okay, so that's a little bit of both. A little hard to interpret, but now we lost the distinction between the rapid initial and final phases, essentially. Okay, and I'm not going to pause to draw. There's some interesting conclusions we could draw from that, but I'm going to hasten on. So here's an interesting point. Well, I'm not going to, it's not here, it's coming up. The interesting point is going to be here. Okay, so now let's do the same thing, but it's the same model, but now we're making it stochastic. So we've added in an element of drift. So now we're going to have drift selection balance. This is the OU process we looked at before, but in 2 dimensions. And I guess the quickest thing, so in red, the adaptive landscape as analogous to a 95% confidence limit. The green circle is, I'm mixing color schemes from animation to animation. Green color scheme shows the G matrix. The blue circle shows the 95% confidence limits for the process, the limiting process at long elapsed time. Okay, so just as in the univariate case, the population buzzed about a band through time. Here we're showing time as an animation, and we've got a circle defining, the circle is a function of both the G matrix and omega plus, the omega plus P matrix. I'm not going to show you that animation because it's just going to show you points buzzing about. So now, here's the point I was trying to get to. When we look at the Ornstein-Uhlenbeck process in multivariate space, this is the expression for the variance covariance matrix as a function of time. And this is a stochastic differential equation, and we're looking at the variance. From that, we can derive the expression for the variance among replicates in their mean as a function of time. And if we look at that, we see a negative exponential distribution expression out here, right? So that means that expression goes to 0 as a function of time, and so after really not very many generations, we're going to get an equilibrium value for the variance, and it's given by this expression. Now notice what's missing from that expression. This is a fact Joe and I like to talk about almost on an annual basis. There's no G matrix in there, is it? Is there? So what that's saying is the G matrix plays no role in the ultimate configuration of the adaptive radiation. Just the shape is entirely determined by the matrix that describes the adaptive landscape, that's omega plus P, and the size of that, in general, ellipsoid or whatever, ellipsoid in 2 or more dimensions, the size is determined by effective population size. The bigger the effective population size, the smaller the ellipsoid. So if models in general turn out to be, if this turns out to be the best model for adaptive radiation, it's almost like we wasted your time in the whole first part of the course learning about the G matrix. We should have promised you this irony when we launched into the G matrix, or even quantitative genetics at the very start. So if you didn't like the G matrix, we just made your day. Well, we can look at other models of radiation and see what's true. Okay, yeah. But it's unlikely that this is going to be our best model. I'm going to show you this quickly later on, and I'm going to talk about this later in the course too. This is the heartbreak of OU, much as people like the OU. When you actually try to account for real radiations, or just the magnitude of radiations, in general, to do that, you'll see this in the case of birds of paradise, trying to account for the evolution of their plumes. To get the OU process, with this limiting value, time's not so much the thing as this limiting value, you need either small n sub e, so you have a very big stochastic point, or you need very weak stabilizing selection, or both. And the values you have to assume seem, I think, unrealistic. I'll give you some examples later. So I love the OU, but a simple OU is going to have problems. We can show you that later, but it's our conclusion for now. All right, so now we've done just about all of the simple, well, not all. We didn't do an exhaustive inventory of possible models with a stationary peak, but we've gone to 2 dimensions now. What this suggests is to account for adaptive radiations, to get an amount of evolution as a function of time that really represents the adaptive radiations that we're familiar with and want to explain, we're probably going to have the peak move. And again, we'll reinforce this point as time goes on. And so here's a model of Brownian motion in which we do that. So we're going to have, so over on this side, I show you in orange the adaptive peak of an adaptive landscape that is moving by Brownian motion. And so here's the simple model for that. Each generation, the position of the peak in 2 dimensions is a function of the position of the peak in the generation before. So this is in one trait. We're in one dimension, sorry. And we take a draw from a normal distribution with some fixed variance. Right? This is like taking a draw from, the drift anyway. So that's a model of Brownian motion, and here's an example of the peak doing. And then here's the model of the lineage, which is now evolving by three processes. First of all, the peak is moving. Secondly, you can see from this term, the mean of the lineage is tending to move towards the peak. Thirdly, the mean is being displaced away from the peak by a drift contribution. Again, a draw from a normal distribution with zero mean and a variance given by the G over N sub E term. Okay? And if you look at this, you can see that at least with these parameters, very small variance in the peak each generation movement. The mean is pretty much tracking, not completely accurately, but it's tracking that moving optimum. And that's what genetic variance does for you. It enables you to track. See, here's the genetic variance term there. So if we do multiple populations, then you get a picture like this. And we have a rather complicated expression for the ever-expanding variance in the peak. Now, I'm not going to take the time to dissect this as we could, but the point is, with this model, with Brownian motion, like with simple genetic drift, which is also Brownian motion, the variance is ever expanding. It doesn't reach some limiting value. And so this can be a model for a potentially good model for adaptive radiation. It's just a question of whether the timing is right, and can you get it to produce the pattern that you're seeing in your adaptive radiation on the time scale you're looking at, with stable, with population size of, in a feasible range, and a genetic variance that's, say varies between 0 and 1, somewhere in maybe with a genetic variance of 0.4 when P is 1. So we're taking the mode of that survey we looked at. Okay, I'm moving kind of fast, but let's stop. Any questions at this point? Trying to get to the data. Sophia. So we're doing an outrageous thing. We're holding G constant at a particular value throughout the radiation. And you're good to notice that, because that wouldn't be literally true. G, as well as the whole G matrix, could be fluctuating. Well, we're just taking the average value and imagining that there could be fluctuations, but it's about some value. But the other thing you quickly learn running these simulations, even on this generation, is the value of G doesn't matter that much. Particularly on a time scale of hundreds, thousands, or tens, or especially on a time scale of tens of thousands of generations, it just doesn't affect the process very much. Tempts one to conclude that we can disengage from the front part of the course, doesn't it? You might consider...
Yeah, if G is 0, it has an effect. If there's no genetic variation, it won't go anywhere. But
Right.
I mean, basically, you're having a peak moving around, and the population is being towed with connected by an elastic band, a rubber
Right.
band.
Yeah.
And for short periods of time, that can be important. But if the peak goes far enough, more or less, the population stays
Right.
close enough to it that most of the motion then is the peak motion.
Yeah, that's right.
That's
right.
That's equivalent to.
So the
Yes.
the process of peak movement is the kind of like the big controlling force. I mean, you can see here that even if we let these expressions go to their limit, there is a contribution of G right here, and so it's not 0, but it's contributing to the variance, but it's not necessarily. Okay, so here I'm going to go right past this. There are other possible motions of the optimum. I'm going to show you a quick model, which we'll use later. It's not a particularly realistic one, but it's a handy one in a theoretical sense. So this is white noise. So each generation, instead of, remember with Brownian motion, we'd have the position of the optimum plus this draw from a normal distribution. Here it's just a draw from a normal distribution. So the peak is bouncing all over the place, right? And when the mean tracks that erratically moving optimum, it does reach a stable limit, and basically you can see the peak is moving, over broad limits, but the mean is moving much less because it's basically damping these generation by generation movements of the peak. Not very realistic that the peak would be moving that much, and if you have really severe movement, then you might put the population at risk of extinction. Well, we'll use this later. I just wanted to give you a slide showing you what we're using here. And here's the replicate lineage version of that. Okay, here's another model we might think about, and in this one, we've actually got white noise movement of the optimum within limits, and then we have a rare Poisson process, and so periodically that Poisson process calls for a jump in the optimum, and then when that jump is called by the process, we take a draw from a normal distribution to specify how far the peak moves. So the point of this is that with this process, we can stay within stable limits. Here I'm showing some limits that we might expect for evolution. I'll show you why I've drawn them exactly there. And with this model, we can stay inside those limits. We can produce a pattern of stasis, but then we can see periodic departures from stasis when the Poisson process pops us out of what I'm going to call an adaptive zone after Simpson. And here are two very substantial movements of the optimum theta, and then long waiting periods, relatively long. I've speeded up the process just for this figure. And the reason I'm going a little bit fast here is you can look back over the details in the model. I wanted to show you some actual data since we haven't been doing very much of that thus far in the course. So this is a data set compiled by Phil Gingrich, a paleontologist at Michigan, University of Michigan. It's longitudinal data, so he had 6,000 values for trait means in intervals ranging from 1 to 10 million generations. A lot of sources, some of them time series, not just one change in the mean. Often these were tree-based. Let's see. Wait a minute now. Well, I'm showing this is. Trying to think if I've got the right. Anyway, so this is actually. Right, this is part of this is Gingrich's data. I'll show you that, but this includes the tree-based data was added by Josef Weda, and it's taken from a tree, and we had to average over nodes to get it. God. So, okay, and so the traits are primarily size, but some heuristic data, and they come from all sorts of organisms, ranging from forams all the way up to dinosaurs like Triceratops. Okay, so a huge data set, and we're going to try and use all of that bulk data to compare it to our models. And that's what I'm going to first of all show you the bulk data, and we're just going to plot it. This is the microevolutionary part of the data. These are data on, you can see now on a year scale, data in which the interval between the sample points is on the order of 10,000 years or less. Okay, let me show you what this means. This means if we took this point, for example, that means if you take the value at the start of the interval and the value at the end of the interval, the mean shifted by, we're calling this body size because they're mostly size, the interval shifted, the mean shifted by about 50% in the downward direction. Okay? And the interval was pretty close to 10,000. So each of the points has that property. It's how much the mean changed in percent percent body size. They're all, think of them as all size related, over some interval of some length. Now, there's a remarkable feature when you look at the microevolutionary data that there's no overall trend, right? No matter how long the interval is, you're still in a pretty narrow boundary. And if you look at the average population, it's very close to zero. So these are strong evidence for stasis. Yeah, Michael. So it is on a log scale, but I put it in percent change in body size just so it's a little more intuitive. Okay? Yeah, I know, but most of our change is going to be hidden here close, and so what we're going to, well, let me show you. Okay, so the point is the predominant result is predominant pattern is stasis in the microevolutionary record, and bounded evolution about that. You don't go very far away. Now let's add in the fossil data. We add in the fossil data, we start to see some points out here like this. But still a picture of relatively bounded evolution. And then when we add in tree-based data, well, I'm not going to add in tree-based data. I'm just going to show the conclusion we'd reach on the basis of this, and that is that if we try and fit Brownian motion, say the genetic drift version to this, what we fail to do is account for this evolution, these points on a short time scale, and we go roaring out into this space much more often than we're seeing in the record. So we get too little differentiation on short time scales. We tend to go like this, and then we go blasting away into this zone where we have very few data points. So with a big data set, 6,000 data points here, we concluded from this that Brownian motion, the genetic drift version, is a very, very poor model. And so are most other models of adaptive radiation. For example, the OU process is not going to account for these relatively rare departures, but they're substantial out there. Okay, now let's add in data from trees. And when we do that, we see really substantial evolution, but on a time scale of 10 to the 8th generation. So that's 1 million years, 10 million years, 100 million years. So especially on a time scale when we get out to 100 million years, we've got huge amounts of evolution possible. But still, the preponderance of observation are in a band that I'm going to arbitrarily draw here at about plus or minus 65% change in body size. So now we're looking for a model that would do that. Simple OU isn't going to do it because it's bounded, right? It's never going to do that. Simple genetic drift is not going to work because it goes roaring out here much too soon with any realistic parameter values. And so I'm going to summarize rapidly now a paper that I did with Josef Weta, Tom Hansen, and Jason Pinar. And here's what we showed. That is, when you do basically a maximum likelihood comparison of models, the best fit is a multiple burst model of that type I showed you before. And so what that does is you get a state. So what we did is we took white noise, that simple white noise model, to try and count for what we call the long part of the barrel of the blunderbuss, and then we're using other processes to try and account for the explosion that we see out there in long time scales. So here's the white noise only model. Doesn't do very well. Brownian motion does actually quite well, but, and I didn't talk about single burst, that's just one jump. But if we have multiple bursts, here's single burst, this actually does quite well. And although it looks about like the same fit as with Brownian motion. What Brownian motion does is predict. It doesn't predict enough evolution right in the center of the enough stasis right in this part. It's not obvious from the figure. So that multiple burst model I showed you makes a nice fit to the data. So let's look at it in detail. We're going to fit it by maximum likelihood, and we can actually estimate the size of the bursts that we require and the interval at which they happen by random chance in a Poisson model. So here are the maximum likelihood fits to those parameters. So if you look at so here's the distribution of white noise and dash, and then here, when the Poisson process calls for a burst, a change in the optimum, here's the maximum likelihood fit for the distribution that fits best, and you can see the burst sizes are not very big. A lot of times you get a burst, but you're still inside what looks like stable limits. The tail, though, will carry you outside, and on a very long time limit, you'll get a succession of bursts, and that can carry you out into this range here. Now, the waiting time between the bursts fit by maximum likelihood is given here, and the mode of that distribution is here at about 25 million years. So there's a long waiting time in general, on average, between these bursts. And I'm embarrassed to say that the four of us did not make this connection. I'm going to describe it. Did you see the obituary for David Raup in the New York Times?
I knew he had died recently. Yeah.
So I knew Raup when I was at Chicago. So I was reading the obituary closely. I'm going, Jack Sapkowski and so forth. Get about to the third paragraph and said, the author of the obituary, Raup is especially known for the discovery that there are periodic departures from the background extinction rate, so that there are profound episodes of extinction in the fossil record at about an average interval of 25 million years. I thought, oh my God, my God, that's what we found. So here's the point. Whether this is true or not, what we're looking for is some kind of ecological explanation for why there's this long wait for jumps in the optimum. And the Raup-Sapkowski result is perfect for us, because if on the average, and this is the right time scale here, on the average you wait 25 million years, there's a big extinction event, and then there's a burst of evolution, and ecological conditions on the heels of extinction might. I'm not saying we have to do a lot more checking. We didn't say this in the paper because we weren't thinking about.
Could you explain to me why?
At this point, it's an interesting coincidence.
Why the dashed line there looks more like 250 million years?
This one?
Yeah, not 25.
A million, 10 million, 100 million.
Oh, so 20. Oh, you're right. 10 million. I'm sorry. I can't calculate exponents in my head anymore. Could you also just so I can rescue myself from that embarrassment, in the white noise distribution, you can't really tell that from a rather rapidly returning OU, can you?
No, I mean, so we're not even serious about. We put in white noise just as a sort of, there's some OU-like process, and we used white noise here.
Because if you sample from an OU that's sitting with the optimum sitting in the same place, there's a sort of return time if you depart, and if you take samples much longer than that return time, they'll look independent, identically distributed, and they'll look as if they're from a white noise process.
Yeah.
You won't be able to tell the difference.
So you're not shocked that we use white noise here. It just doesn't matter.
Just annoyed.
Yeah, just annoyed. Okay. Yes, Matthew. I have to not.
Perhaps this is obvious. I'm looking at that picture, and if you had erased that dashed red line, is it obvious where it should go? Is that 25 million years?
This is the mean time of our distribution fit by maximum likelihood.
Okay, so
Model.
it's not going to be apparent just from looking at that plot there.
Well, this is the mean of that.
Well,
of that
yeah,
distribution.
that plot, but the one above.
Oh, they're on the same scale. You know, stretched so that this lines right up like that. You like?
Okay, what? Yeah, if I was just looking at that top plot, and all the red lines were, and that red line was.
These are the data. This is the model that we're trying to explain them with, right?
Okay.
Yeah. No, it's not obvious.
Okay, I wasn't sure if there was something
No, so
obvious that I was
no,
missing.
No, you're right.
Once I see the bottom, I can see it.
Question, were we done? Okay. I think it was my goal to irritate you a little bit by going fast, but to be inside my time. I'm not too bad. We have 20 minutes for discussion if I ended right now, and I'm almost done. Oh yeah, look, okay, I'm just about done, okay. Okay, what have we learned? Quantitative genetic models can be used to at least produce candidate models of adaptive radiation. Models in which the adaptive peak is stationary cannot account for the scope of actual phenotypic radiation. And I just sort of glossed over that, but we'll talk about that. We'll talk about that later. I just can't find a model of a stationary adaptive peak that accounts for the full scope of adaptive radiation on long time scales. And so when we look at actual data sets plotted on a time scale, bounded evolution then is the prevalent mode of evolution. And this is what a lot of, you want to think about punctuated equilibrium. But actually Simpson and Darwin, long before Gould and Eldridge, were pointing out that this is the predominant mode of evolution. And Simpson talked about the fact, so let me give you an example. I got a little bit, let me expand a little biology. What Simpson is saying, what Simpson said is if you look at radiations of mammals, like for example, let's take gophers. If you look at gophers, let's look at weasels, it's an even better example. If you look at the radiation of weasels, the amount of evolution is constrained so that weasels are evolving within a set of limits, and they look generally like bigger or smaller versions of one another. A mustelid is a mustelid. And all features of their body are pretty highly conserved, and there's radiation in body size. But what Simpson is saying is the mustelid way of life, general ecological way of life, is preserved on a tens of millions of years time scale. And he's talking about then, and he draws pictures that look very much like our picture of bounded evolution, in which there are boundaries, as Simpson calls them, the edge of adaptive zones, and not just speciation, but whole evolution and extinctions of genera within evolving families are happening inside that zone. And so that's what he's describing is bounded evolution that is only very rarely breached by the evolution of a new family and a new adaptive zone. Yeah, you wanted to comment, Felipe? We have a microphone for you.
So isn't it possible that some sort of circular argument could be happening there, because we a priori define species as organisms that are already similar or as similar as possible, right? So like the closest two species are the more similar they are. So I'm just wondering if the comparisons of the pairs of species that are being compared to get the difference in body size, that small difference is because of the definition of like closely related species being more similar.
So when we're out at the blunderbuss, the expanding part of, we're talking about the origin of new genera, right?
Okay.
Okay.
Uh-huh.
We're far beyond far beyond speciation.
Okay, that's nice.
Let me back up. This is a point Simpson made. 2, go back here. Most vertebrate speciation is happening in this part, right? Out, on a scale of maybe 1 million to 10 million years out here. This whole part out here, this is the evolution of new genera and families. And that's when speciation is not getting you really it's not accounting for evolution outside these boundaries.
Okay, I see. And is there any maybe that's the tree of life data, but I'm not, maybe I didn't follow.
This means phylogeny.
Like if how will that graph look if you take comparisons of a lineage? So like.
Yeah. So let me back you up. So all of these data are lineages.
Okay.
Not necessarily a single species. They might be lineages that transition into, make it one or more species transitions. But most of this is within genus evolution.
Okay, nice.
Okay, does that help?
Yeah.
But when we do this, that now at the end of the, there are some species that have been around for a lot of time, and they're down in here. So it's not that everything is the origin of new genus, but this stuff out here, a lot of that is.